Hello everyone, I am Vaishnavi Sapka. I am a student of Ashok Ramani Institute of Pharmaceutical Sciences and Research Save, subject Pharmaceutical Microbiology, guided by Mr. A.N. Dhage. Introduction. Sterility testing is a critical process in pharmaceutical manufacturing. Ensure the absence of viable microorganisms in pharmaceutical products. IP guidelines provide a framework for sterility testing. Sterility testing involves first sampling, taking representative samples from the product batch. Second, test method, using standardized test methods such as membrane filtration, direct inoculation, lyophilization. Third, incubation, placing the samples in a controlled environment, for example temperature, humidity, to allow microorganisms to grow. 4. Observation, monitoring the samples for sign of microbial growth such as turbidity, colonies, or other visual indicators. 5th. Interpretation, evaluating the test results to determine if the product is sterile. Number of containers in the batch, minimum number of containers recommended to be tested. First, parenteral preparation, not more than 100 containers, 10% or 4 containers, whichever is greater. More than 100 but not more than 500 containers, 10 containers. More than 500 containers, 2% or 20 containers, whichever is less. For large volume parenterals, 2% or 10 containers, whichever is less, unless otherwise justified and authorized. Second, ophthalmic and other non-parenteral preparation, not more than 200 containers, 5% or 2 containers, whichever is greater. More than 200 containers, 10 containers. Third, surgical dressing and devices, catgut, surgical sutures, and other sterile medical devices for use. 2% or 5 packages, whichever is greater, up to maximum of 20 packages, not more than 100 packages. 10% or 4 packages, whichever is greater. More than 100 but not more than 500 packages, 10 packages. More than 500 packages, 2% or 20 packages, whichever is less. 4. Bulk solids, up to 4 containers each containers. More than 4 containers but not more than 50 containers. 20% or 4 containers, whichever is greater. More than 50 containers, 2% or 10 containers, whichever is greater. Methods for sterility test. Method A, membrane filtration. Method B, direct inoculation. A. Membrane filtration, suitable for the substance like oil and ointments that can be put into a solution, a non-bacteriostatic solid not readily soluble in culture medium, a soluble powder, or a liquid that possesses inherent bacteriostatic and fungistatic properties. Method A is suitable for liquid product where the volume in the container is 100 ml or more. This apparatus is used for the membrane filtration unit. Membrane filtration apparatus for sterility testing. Closed reservoir and container to collect the filtrate between which a properly supported membrane of appropriate porosity is placed. Porosity is about 0.45 micrometer. Diameter is about 50 millimeter. Flow rate is about 55 to 75 ml per minute. Pressure is about 70 millimeter mercury. Cellulose nitrate membrane for aqueous, oily, and weakly alcoholic solutions. Cellulose acetate membrane filter for strongly alcoholic solutions. Dilution fluids. Mainly 2 types of dilution fluids are used. Likely fluid A, which is prepared by dissolved 1 gram of peptic digest animal tissue in 1 liter of water. Filtered and pH adjusted 7.1. Poured the fluid into 100 ml of flask and then sterilized at 121 degrees Celsius for 20 minutes. Fluid B, which is prepared by using fluid A. To each liter is added 1 ml of polyabsorbate AT. This is prepared if the sample contains lecithin or oil. Then pour the fluid into 100 ml flask and then sterilized at 121 degrees Celsius for 20 minutes in autoclave. Quantities of sample. First, for injectable preparations. The uses of the contents of the containers are listed in below. Quantities of injectable preparation for sterility testing. Quantity per container. Minimum quantity to be used for each medium unless otherwise justified and authorized. For liquids, less than 1 ml, whole contents of each container. 1 to 40 ml, half contents of each container but not less than 1 ml. Greater than 40 ml and not greater than 100 ml. For 20 ml. Greater than 100 ml, 10% of the contents of the containers but not less than 20 ml. For solid less than 50 mg, the whole contents of each containers 50 mg or more but less than 300 mg. Half the contents of each containers but not less than 50 mg. 300 mg to 5 gram 150 mg. Greater than 5 gram 500 mg. Second, for ophthalmic and other non-injectable preparations, the uses of the contents of the containers are listed in below table. Quantities of ophthalmic and other non-injectable preparations for sterility testing. Type of preparation, quantity to be mixed, quantity to be used for each culture medium. First, ophthalmic solutions, other non-injectable liquid preparation 10 to 100 ml, 5 to 10 ml. Second, other preparation, preparations soluble in water or appropriate solvents, insoluble preparations to be suspended or emulsified duly. For example, creams and ointments 1 to 10 gram, 0.5 to 1 gram. Absorbent cotton not less than 1 gram. Methods of sterility testing. First, for aqueous solution. Second, for liquid immiscible with water. Third, for oil and oil solution. Fourth, for ointments and creams. Fifth, for soluble solids. Sixth, for sterile devices. B. Direct inoculation. This method is used for small volume samples. The volume of the product is not more than 10% of the volume of the medium. It is suitable method for aqueous solutions, oily liquids, ointments and creams. The method involves aseptically opened each sample container from a sterilized batch of products. Methods of test. First, for aqueous solutions and suspensions. Second, for oils and oily solutions. Third, for ointments. Fourth, for solids. Fifth, for sterile devices. Interpretation of results after incubation and during incubation period. If growth is not observed, if growth is observed, if growth is not observed, passes the sterility test. Preparation sterile. If growth is not observed, sample passed. Container, if growth is observed, containers are reserved and retest is performed as in the original test. If growth is observed, if they are not readily distinguishable from those growing in containers reserved in the first test, preparation fails the test. If they are readily distinguishable from those growing in containers reserved in the first test, secondary test is performed using twice the number of samples. If growth is not observed, preparation passes the test. If growth is observed, preparation fails the test.